Hello students, welcome to Raaonline. The chapter is genetics, that is inheritance and variations. Class 12th NCERT biology book. And this topic is discussed here in brief with question. For some simple questions are there in this topic also. For the crash purpose. So first question is with regards to the Dandel. Actually, I would like to give the brief idea about the genetics. Genetics, science of biology, branch of biology. Genetics is branch of biology. This term was used first by W. Bateson. This term was given by Bateson in 1906. This is question for medical entrance. And genetics is the study of heredity and variation. Genetics is the study of heredity and variation. This is question number 2 for the examination, already asked in the exam. What is heredity? That is transmission of parental characters from one generation to next. That is a statement is generally given for this, that is like begets like begets like. I mean offsprings resemble parents, and what is the cause behind it? That is heredity, transmission of parental characters from one generation to next. And variation, that is two offsprings of a parent, if they are not identical twin, provided if they are not identical twin, then they have some identifying feature, that is they are not very similar. This is due to the variation. This is the cause of variation. So what is variation? That is two progeny of a offspring, two progeny of a parent, they are not fully identical, that is this is the variation. And a study of heredity and variation is dealt under genetics. And genetics without Mendel is not possible. Mendel was the pioneer of genetics actually. Gregor Johann Mendel. Mendel was pioneer of genetics, that is his full name is Gregor Johann Mendel. Gregor Johann Mendel, born in 1822 and died in 1884. Died in 1884. That is Mendel worked in 19th century. Mendel worked in 19th century. Mendel worked in 19th century. Mendel experimented over pea plant, garden pea plant. Mendel conducted several hybridization experiments on garden pea. Scientific name of garden pea is Pisum sativum. Member of family Fabaceae. Mendel conducted number of experiments over garden pea. So one question is also possible, why did Mendel select garden pea? Then it was already described in my theory lecture, and here I would like to give in the brief, that is presence of several contrasting traits. Presence of several contrasting traits. Second, bisexual plant and predominantly self-fertilized, can easily be cross-fertilized. This is the second advantage of a garden pea. Third advantage, that is it's a seasonal or annual plant, so results can be obtained in shorter duration. And it can easily be cross-fertilized, that is it can be easily be fertilized, that is responsive plant. But Mendel died as unknown fellow. In 1900, this is the year of the rediscovery of Mendel's work. Sorry, beginning of modern genetics. And in this year, his work came in limelight. Mendel's work came in limelight, and the three fellows, that is De Vries, Correns and Sherman, these three are the rediscoverer of Mendel's work. It means one is out, that is Muller. Muller is not involved in the rediscovery of Mendel's work. Mendel's work was rediscovered by number 1 the Vries, number 2 the Karl Correns of Germany, number 3 Austrian the Sherman. Karl Correns was a person who compiled Mendel's work. Compilation of Mendel's work was done by Karl Correns and formulated his work. He formulated Mendel's work. Formulated Mendel's work presented in the form of laws. Presented in the form of laws. So these three are the discoverer in this question. Now come to the second one. The gene controlling 7 P characters, 7 characters of P, studied by Mendel, now known to be located on how many of different chromosomes? They are located on 4 chromosomes. The traits studied by Mendel, or character studied by Mendel, they are located over the 4 chromosomes. Mendel studied total 7 characters, and they are located over the 4 chromosomes, and he studied one particular character at one time. Suppose he studied height, then nothing is considered at that moment. He studied only one character at one time. So total 7 characters Mendel studied, and each one is with 2 variants, that is 7 pairs of contrasting trait. We can say in terms of trait 7 characters but 7 pairs of contrasting trait. Mendel studied 7 characters. Or 7 pairs of contrasting traits. 7 pairs of contrasting traits. Seven pairs of contrasting traits. Right? And their location is on 4 chromosomes. And these 4 chromosomes are chromosome number 1, chromosome number 4, chromosome number 5, chromosome number 7. These are the chromosome numbers on which Mendelian's 7 characters were present. And on chromosome number 1, only 2 characters. At chromosome number 4, 3 characters. Chromosome number 5 has 1 character, and chromosome number 7 has 1 character. So maximum characters are located over the chromosome number 4. 1, 4, 5, 7 is chromosome number, and this is the number of characters present on them. On first chromosome 2 characters, on fourth chromosome 3 characters, on fifth and seventh chromosome 1 each. So for this question, the answer is on 4 chromosome, question BHU, medical entrance. This was already asked in the various exam. One of the prestigious exam was BHU. Now it is merged in the NEET. BHU PMT, Banaras Hindu University PMT. It was already asked this. They already asked this question. And this analysis was actually made by Blixit. Blixit made this analysis. The main reason for the success of Mendel, he made a statistical analysis of the right. His first book he first took only one character at one time, right. He kept pedigree record, no. He selected pea plant. Out of these two, which one is the best? Actually, first two, these two are the not these two are not related with the success of Mendel. The first one is comparatively better. Why? Because statistical analysis was made by the Mendel only. Proper mathematical approach was taken by the Mendel to describe his result. Before Mendel, number of persons also performed hybridization experiment, but they are not able to provide proper result. And Mendel was successful because of its statistical analysis or mathematical approach. And also one character at one time, but this is comparatively better. Pure line means homozygosity. What is homozygosity? That is, if a trait is controlled by identical gene, either AA or small aa. These are the factors or alleles. If a trait is controlled by two identical allele and in an organism, if that trait is controlled by the two identical allele and if it is selfed, it produces same type of individual, that is pure line. So here the question, that is pure line is homozygosity, homozygosity with one dominant gene, no, and heterozygosity for one linkage. Better answer homozygosity. Is homozygosity. That is, if it is selfed, it produces identical progeny. It produces identical progeny. It may be AA or it may be small a, but the identical allele should be there to control that particular feature or trait. One of the contrasting form of a trait is known as allele. So simple answer, that is allele. Alleles are the variant actually. One of the contrasting form of a character's term to be, that is alleles. Allele term. Again the allele term was used by Bateson. Term allele was given by Bateson. That is alternative form of a gene. It may be A, it may be small a. That is allelic combinations, either AA, either capital A small a or small a. These are all allelic combination. That is alternative form of a gene, that is allele. It may be identical, it may be unidentical, that is allele. One of the following is correct statement. Dominant trait is expressed in the homozygous condition. Dominant trait cannot be expressed in heterozygous condition. Recessive trait can be expressed in homozygous condition only. Recessive trait can always express in the heterozygous condition. Let's see. We have studied the genetics in 10th as well as in 12th class. And while performing Mendelian, we are studying the Mendelian experiment. One variety is tall and other variety is dwarf, and we write the genotype in this manner. And if the cross is made between these two, F1 variety is all tall. It is again tall. See this one. Here tallness is controlled by two identical capital T. And here this plant is also tall, but the identical alleles are not there. I mean, this plant is tall because tallness is dominant over dwarfness. I mean to see, it means that is dominant gene expresses itself in homozygosity as well as in heterozygosity. But you see, even at this level, we can say the recessiveness a recessive trait is expressed only in homozygosity. Recessive trait is expressed only in homozygosity. Suppose a cross is made between hybrid tall plants, that is selfing between hybrid tall plant is done, then you see the capital T, small t, gametes of one parent, and then capital T, small t from other side, that is male and female gamete. T, capital tall, sorry, capital T, small t, this is also tall, capital T, small t, this is also tall, but dwarfness, for dwarfness, that is homozygosity is required. I mean to say dominant gene, that is capital T, expresses in homozygous condition as well as in heterozygous condition, whereas recessive allele expresses only in homozygosity. Because you see in this combination small t is there, but still plant is tall because its feature is dominated by the other one. So here in this case the answer is recessive trait is expressed in homozygosity only. This is the result of this question. Which one of the following pair is consist of both recessive traits? In our NCERT book, total 7 characters and with dominant and recessive trait, a list is given. That this question is from that particular list, and dominant trait is that trait which is expressed in the hybrid, and the trait which is not shown by the hybrid is termed as a recessive trait. In first case, dwarf plant, it's a recessive, but axial flower is dominant. So this is the wrong answer. Constricted pod, again it is a recessive trait, but round seed is dominant. So this is also cancelled. Green color of seed coat and terminal flower. This is correct. This is correct. Green color of the seed and terminal flower. Green dominant over yellow. Sorry. In case of seed, yellow is dominant over the green. In case of seed, yellow is dominant over the green. So green seed color is the recessive trait, and terminal flower is also recessive. In flower position, axial is dominant actually. Ninth question. 2 crosses between the same pair of genotype or phenotype in which the source of gamete are reversed in one cross, that is reciprocal cross. This cross is known as reciprocal cross. Why reciprocal cross? One plant is tall, capital T, and other plant is dwarf, small t, and this plant produces bisexual flower, and this is also produces bisexual flower. If this plant is treated as male, then the source of male gamete is T. That is, this plant is treated as male in one cross and other one treated as female in other cross. Suppose we mention as A and B. From A, male gametes are taken in this cross, and from B, female gamete. During the second cross, now A is treated as female, and B is treated as male. That is, source of gamete is reversed. That is what? Reciprocal cross. Superiority of hybrid over the parent. This is the pre-medical question again. This is called heterocyst or hybrid vigor. This is called heterocyst or hybrid vigor. This is hybrid vigor or heterocyst. It is also called hybrid vigor. Term heterocyst is given by G.H. Shull. G.H. Shull. That is, hybrids are better than pure. I want to give you the one very good example that is associated with your 12th book, and it is seen in which section? That is strategies of food or plant breeding section. Sugar cane. One example, sugar cane is there. The sugar cane in the northern area, Saccharum barbarii, it has weaker stem and shorter and low sugar content, whereas the sugar cane of south, that is Saccharum officinarum. In both the cases, that is sugar cane of south is have it long and thick, long stem and thick stem. So sugar content is more, but it is not suitable for the northern area. In that case, the cross is made between these two. That is southern variety of sugarcane and northern variety of sugarcane. A variety is generated and that variety is fit for that particular environment, that is northern India. It can be easily be grown in North India with proper feature, that is first of all suited to the North India also, then having long stem and long and thick stem, that sugar content is also good and the better fitness in the environment. This is the result of cross. So hybrids are always better than pure. Heterozygous tall. Let's come to the let's make the genetic makeup, that is capital T small t. This is heterozygous tall. If it is crossed with the homozygous dwarf, that is a small t, because dwarf plant is recessive and recessive parent expresses in homozygosity. Then what proportion of the proportion of dwarf plant? This plant produces two types of gamete, capital T, small t type, whereas this plant produces only one type of gamete, that is a small t. You can write either one t or you can write both t, no problem. In both the case, result will never change. Result will be same. Capital T, small t, capital T, small t, last one small t and small t. This checkboard display system was introduced by R.C. Punnett. One more information, that is checkboard system was introduced by R.C. Punnett. So that's why this checkboard is called Punnett square. This checkboard display system is known as Punnett square. This is question of NEET hardly 2 years or year or two. Punnett square, he was British. R.C. Punnett was British. Ab see, let's analyze the result. That is tall plant, dwarf plant. First two tall, that is out of 4, 2 are tall. That is 1 by 2 tall. Similarly, see these two plant out of 4, they are dwarf, that is 2 by 4 are dwarf. 2 by 4, that is 1 by 2. 1 by 2, that is 50% dwarf. Question says what proportion of dwarf, that is 50% or 1 by 2. 50% or 1 by 2 here. So answer is 50%. This is the answer of this question. A pure tall plant, see this one, language, pure tall and pea plant, that is tallness is dominant and it is pure, that is both the alleles are identical. They are raised in the nutrient deficient medium and due to which they become dwarf. due to which they become dwarf. Right. If such plant is selfed, see this one, genotype doesn't alter. Genotype does not alter. The genes for the tallness remain in their plant, but they are not expressed because of nutritional deficiency. So if such plant is selfed, the plants will be, if the conditions will normal, then plants will be tall. There is no change in the genotype. If they are their progeny, because they are not genetically dwarf, they are physiologically dwarf, they are by means of the physiological fitness they become dwarf in deficient medium. If they get the normal media, genotype does not alter, they will be tall. They will be tall. That is, such tall plant, dwarf plants will also produce tall plant because gene in them is for the tallness. Genetic makeup does not alter. That's why by their selfing in normal media, they will produce normal tall plants. Mendel's law of segregation is applicable during fusion of gametes. For fusion of gametes, formation of zygote, better word is formation of gamete. How? Capital T plant and a small t plant, this is hybrid tall plant. This is hybrid tall plant. When this plant produces gametes, then in one gamete, gene for the tallness will be there, in other gamete, gene for the dwarfness. That is, these two Mendelian factors for the height get separated at the time of gametogenesis. Or we can also say that is segregation of Mendelian factor. Segregation of Mendelian factor takes place during meiosis. That is, segregation of Mendelian factor takes place during meiosis.